everyone. It's a great honor to present my lab's research in your seminar series of the newly established plant breeding center at the University of Minnesota. It's very exciting. And I hope that by the end of the presentation, I've convinced you that the great work by graduate students and postdocs in my lab are leading to plant improvement, which is a theme of your seminar series. So let's delve right into it. It's to no surprise to you in the audience that we do need to increase the productivity of crop plants. And that's because the world population continues to grow and is expected to reach 10 billion people by 2050. And in order for food production to keep up, we do need to increase that by 60%. And that is due to the loss of agricultural lands, more agriculturally marginalized environments, as well as the impact of climate change. And so how to increase the productivity of crop plants, or one way to go about, or the multiple ways we could go about it. This was something that was discussed about 10 years ago at a meeting in Asilomar. And a summary of that meeting was published in Nature, entitled Feeding the Future and led by Susan McCouch, where we talked about how we could improve crop productivity with a major focus on resources that is available in stock centers. And it's there where I got the idea of the research that I'm going to present to you today. So that started, or that seed was planted, no pun intended, started about 10 years ago. So one way to address crop productivity is to tap into wild relatives and semi-domesticated germplasm, and particularly the germplasm that's already stored in our seed banks across the world. So the target traits for crop improvement, of course, it's no-brainer. It's biotic and abiotic stress resilience is incredibly important because they're directly related to yield. And crops are now grown worldwide and not in the areas where they originated. And so plants, of course, adapt to their ecological niches. And adaptation is the greatest in the place where the species originated. And that's why resistance to stress, and whether it's abiotic or biotic, are often found in wild relatives of cultivated plants in the locations where the species originated. And to just kind of segue a little bit into where those centers of domestication are, this was discovered or first described by a very brilliant scientist, Nikolai Vavilov, who traveled the world and collected germplasm from various places and brought it back to Russia. And he just wrote in this book that was published almost 100 years ago that where the primary centers of crop domestication were. And of course, it's the usual suspects. We know that now, but back then it was new. The rice was domesticated in Asia. Here's the area that encompasses the Fertile Crescent, where wheat, barley, and oat were domesticated. And in the Americas, of course, in South America, this is the area of domestication for potatoes. And in Central America and Mexico is where corn was domesticated. So that's where you would find the largest diversity, as well as the largest likelihood of alleles that are adapted to the climate and the diseases that are prevalent in the areas. There are other traits that are important for crop improvement that primarily arose during domestication, either because they were selected due to standing variation in wild relatives or to the newly arising mutations. And these are traits that are not so easily found in wild relatives because they don't display themselves so well. But these are traits related to yield, quality, and reproductive development. For yield, of course, you have plant architecture. Corn is a great example. Selections for certain architectural traits have led to that we can plant corn at much higher densities, and therefore the yield per acre will increase. Produce size is very important. Seed set and fruit set without proper fertilization is important because then under adverse weather conditions, you still have a product. And mitigation of physiological disorders that are often associated or not all, they're occasionally associated with yield related traits. I'll say more about quality in a moment, but the optimal reproductive development is also very critical. It's a proper flowering time for plants. Species that were originated at the equator, if they're grown now at higher latitudes, they would have to adapt to the change and the seasonality. And it's a great example of that is potatoes. Potatoes are from the equator, but to grow productively in Minnesota, as well as other places in the world, they have to really adapt. They have to adapt to the proper flowering time so that they actually tuberize at the right moment. So for the presentation today, I'm gonna say something more about the quality traits and what we are doing in this respect, particularly about produce morphology or size. I'm gonna talk mostly about fruit size. I just want to put in a plug. I'm not going to show any data about the flavor and aroma project that I do in collaboration with Denise Thiemann at the University of Florida. And hopefully this work will be continued soon. So now let me introduce this work. We're waiting to hear from the funding agencies. Now, let me introduce the team that I've been collaborating with in the last 7 years. And it's a large team funded by a plant genome grant from the National Science Foundation. I'm going to highlight the folks that whose research I'm going to present today. And that is the university, the Polytechnical University in Valencia in Spain, that's headed by Maria Jose Diaz. She runs the germplasm center there called COMAF. And the germplasm that we use in our experiments were curated by her and selected by her. Joaquim and Jose are geneticists and have contributed greatly also to the bioinformatic analysis. I'm going to share with you some findings from Ana Casedo's lab. Here's is Ana and her postdoc, Hamid Razifard. I have a picture of him here. And they did a lot of the evolutionary research on the history of tomato domestication. I mentioned briefly Denise. This Denise and the postdoc Libby Frick, and they worked on the volatiles. Again, I'm not going to tell you too much about the volatile research at this point. All of our work is actually, and all our findings are on the solgenomics.net website, or SGN. That's led by Lucas Muller and Nama Menda. Nama is in the picture as well. And that's we even have our own page on SGN, the Varitome page. So everything related to the research you'll hear today, you can find there. And then, of course, my own lab, a lot of the excellent work. Well, it was excellent because we're still benefiting from it. It was done by a very great graduate student, a master's student, Alexis Ramos, and he is in this picture here, who collected a lot of the phenotypic data that I'll share with you today. Alexis's work was continued by Lei Zhang and Lara Pereira, two postdocs in the lab. Here's Lara and here's Lei, and continues as well by graduate student Manoj Sapkota and Chen Feng. So what is the Varitome collection? It's as diverse as we can get in tomato, we think, and it consists of 20 Solanum pimpinellifolium accession. These are fully wild relatives. 120 or 119 Solanum lycopersicum cerasiformi, or SLC, and about 27 Solanum lycopersicum or SLL, and that's the fully domesticated tomato. So our focus in this project was not so much on the wild. There are more pimpinellifoliums around in various stock centers, but it's the SLC collection that we really wanted to tap into, because we assume that is the semi-domesticated collection, and we want to focus our research on that. And so there are other projects going on that focus more on fully cultivated tomato, especially in Europe, but our focus was really on the predecessors of the cultivated tomato. So what's SP, or Solanum pimpinellifolium? Makes beautiful long inflorescences with multiple flowers and tiny fruits that are about 1 gram. They're pretty uniform in color. Most of them are red, although we do have 1 or 2 yellow types, but they're definitely very small in size. The SLC is far more diverse. You get fruit sizes that are as small as the fully wild, the SP, as well as large as fully cultivated. So here's an example of a cultivated tomato, and right next to it, an SLC. So one of the major outcomes from the research by Hamid Razifard in Anna Casedo's lab is with these genome sequences of all the accessions in the Varitome collection, he was able to do molecular dating, which we all know that's not always extremely precise, but still could be very insightful. And so what he found is that the last subpopulations of SP from northern Ecuador and the first subpopulations of SLC found in Ecuador shared a common ancestor about 78,000 years ago. And that implies that SP, the Solanum pimpinellifolium, cannot be the direct ancestor of modern tomato, because agriculture didn't start until, about 60,000 years later. So, or 70,000 years later. So the timing, the molecular dating suggests that pimpinellifolium cannot be the direct ancestor of cultivated tomato, and instead it's likely an ancestral SLC. So with the genome sequences, Hamid was also able to generate phylogenetic trees. So again, this is based on whole genome sequence, and identify the various subpopulations that we find in the fully wild species, SPs here on the tree, followed by SLC from Ecuador, then SLC Peru, SLC San Martin, which is an area in Peru. Then there's SLC collected in northern South America, Central America, and in Mexico. And so we call it also the transition subpopulation, and then SLC that are clearly founded here in Mexico. And last but not least is the SLC. So, so this is the phylogenetic analysis of all these accessions. And then you can, of course, calculate diversity parameters as well. And so based on that, it's clear that we know that the fully wild are highly diverse, but so are actually the SLC from Ecuador and the SLC from Peru. And so this became then our target to look for improvement traits in our project. So with the genome sequence and the molecular dating, we can, with the sequences, basically, we can estimate the evolutionary history of tomato. And this is a figure from Hamid's paper from 2020. And this was the analysis led to the following interpretations that tomato, certainly SP evolved into SLC in Ecuador, where it attained quite large size. And then tomato migrated through Colombia, through Central America to Mexico, where it then eventually evolved into a fully cultivated tomato. Well, more recently, Jose Blanca and our colleagues in Valencia presented another hypothesis. And they did not do whole genome analysis, but instead they divided the genome up in haplotypes of about 500 kb and only in the euchromatic regions of the genome, and established the evolutionary history of each haplotype or haploblock alone. And their interpretation is a little bit different. In fact, they propose that SP may have started migrating north, but regardless, it was evolving into SLC, where it fully became SLC here in Mexico. Then the SLC from Mexico traversed back to South America, where with extensive gene flow with pimpinellifolium, the wild pimpinellifolium here, it resulted in this large size, and that fully cultivated tomato is a direct ancestor of the SLC from South America. So why both interpretations are interesting is if you look at this subgraph here, the fruit weight increases in these subpopulations. Here are the 3 pimpinellifolium subpopulations, and this will come back in another slide. They make fruits that are very tiny, about one, maybe at most 2 grams, mostly around 1 gram. And then the SLC in Ecuador, their fruits are much larger in size, and some are even as large as fully cultivated tomato. And so in South America, the fruit size has increased, but then in Central America, the fruit sizes of these SLCs have decreased again. And that was the reason why, one of the reasons why Hamid suggested that maybe tomato underwent a de-domestication event. It became more feral. But however, the other interpretation from Jose Blanca, suggesting that SLC from Ecuador and Central America is actually more related to SP, is also plausible, especially if we look at the results for the fruit weight. Okay, regardless, what do I'm using somebody else's evolutionary inferences to decide how we can harness this knowledge for crop improvements. And so the key question that we wanted to address is, were any alleles left behind? Is that possible? And the main focus again in my lab is on the morphology of the fruit. And for this particular project, we are most interested in the changes in fruit size. So what's the importance of produce morphology? Just a quick segue again. Most of you might not realize, but it is quite important because the overall shape and size of the produce defines the market class. Is it going to be used for fresh, processed, or spice? Spices for tomato. Tomato is not a spicy crop, of course, but you can imagine if you envision peppers, there's the different types and the different uses are very much dictated by the overall shape and size. Shape and size are also very easily recognized by consumers, and therefore they can define new market niches or maintain existing market niches. Not only for consumers, but also for harvesters. So tomatoes that are harvested mechanically, they do need to have the proper dimensions. They cannot be too small, they cannot be too spherical. And so it's very important depending on the harvesting methods. And the internal structures of a fruit, this is particular for tomato, they also dictate what kind of uses you could have for the tomato. So what are the components of fruit weight? Fruit weight in itself, weigh the fruits, simple. We typically weigh 20 fruit together, and then we get a total weight for 20. But we wanted to be a bit more sophisticated in an effort to hopefully identify genes that would, function at a particular stage or a particular aspect of growth. So one way to demonstrate that, so this is a cross-section of the fruit, maybe some genes function on the pericarp area alone and they enlarge the pericarp, while the rest of the fruit, like the internal part, the columella and placenta, stay the same size. So we can measure now the area of the pericarp. We do that with a software program we developed many years ago called the Tomato Analyzer. Septum and pericarp. So the septum are here, these walls, and again, placenta and columella. And then also looking at one more detail is the level, at the cellular level. So we take a cross section of the pericarp here, and that's shown here in this part of the slide, where you can actually start counting the cells from the outside to the inside of the fruit, and that's in the abaxial or adaxial direction of growth. We can also investigate the cell size in images like this. And using the average cell size with the circumference of the fruit that we get from Tomato Analyzer, we can then calculate the cell number circumference, and that would be a growth in the medial lateral direction. So why we sought out these to get these fruit weight related parameters was that we are hoping to identify genes that affect only one aspect, like only cell number or only cell size, as is the case for some of the known genes. Now, you have to keep one other thing in mind, is that the developmental parameters that lead to increased fruit weight, and they can start as early as in the floral meristem. And here's a tiny little floral meristem here, and then the flower bud in the back that you can see. But already at that level, there's changes occurring that can impact the size of the fruit. And so these processes take place after floral initiation and during ovary development, or at the time of anthesis and pollination during fruit development. And again, these two cellular processes are most critical: cell enlargement and cell proliferation. So I don't want you to read all these details, although I guess you can pause the video and look it over, but it's also in this paper that we published last year. Lara put this very nice together. So this panel here with fruit weight I showed you earlier. The pimpinellifolium fruits are very small. The SLC from South America are larger. SLC from Central America and Mexico are smaller again, but the largest fruits are found in the SLL, fully cultivated. No surprise. What we saw is basically that the septum areas, columella areas, and the pericarp areas show a very similar pattern. If we look at the ratio of the pericarp area, for example, that's the area of the pericarp over the total area of the fruit, you see there's not even any difference from pimpinellifolium to fully cultivated. And what it basically says is that as the fruits are larger, the pericarp's getting larger too, but the pericarp is not preferentially getting larger. So fruit size increases basically act on all parts of the fruit. Similarly, for the numbers of cells, pericarp cell layer and pericarp cell size, you kind of see a pattern that very much mirrors that of the fruit weight. So now this is one more aspect of development. So is larger fruit weight accomplished mostly before anthesis or after anthesis? What this slide shows is what happens after anthesis. So what you can see, what is in this panel, you see the number of pericarp cells, the cell layer at the ovary. So let's say it has 10 cells, and then in the fruit it has 12, and then you get a ratio of about 1.2 or something like that. And so SP, there is certainly an increase in cell layer from the ovary to the ripe fruit. That's what we expected, because we knew that from the literature. But what you see here too is that for SLC Ecuador is actually a much greater increase. So there's more pericarp cell layers than that of found in pimpinellifolium. For cell size, the increase is even larger. So SP cell size increases about 100-fold from the ovary to the mature fruit. But in SLC from Ecuador, you get an increase that can even go over 600-fold in cell size. So certainly, larger fruit weight is accompanied by more growth after anthesis, both in cell number and cell size. It's also before anthesis, but I don't have that on this slide. But this is actually more dramatic. So what do we know? We know of fruit weight genes that have been cloned. Certainly, our lab contributed to that. And there are 5 genes that are cloned, 3 that are real fruit weight genes, they affect the weight of the fruit. So of course, our famous fruit weight 2.2 or cell number regulator. Fruit weight 3.2 is an ortholog of Arabidopsis gene CLU, and we call it SL CLU, tomato CLU. And fruit weight 11.3 is a cell size regulator. So these genes impact the size of the fruit, but what also impacts the size of the fruit that I was alluding to a little earlier are the genes that function in the meristem, and those are LC and FAS. and LC is an ortholog of Bushel, and FAS is an ortholog of clavata3. So these 5 genes together impact fruit size. And here you see the different subpopulations, the 3 pimpinellifolium subpopulations, the SLC subpopulations, and last here is the fully cultivated. And in purple, you see the presence of the derived allele for these genes. So the first 3 bars are for fruit weight 2.2, 3.2, and 11.3, and the last 2 are for LC and FAS. And with the exception of LC, that is found in 1 or 2 pimpinellifolium accessions in our collection, the other derived alleles are only found in SLC and to various levels. But you see that once you get to fully cultivated, that most of the derived alleles are actually present. So what's the impact of these alleles on fruit weight? That's shown here on the right. And so if you, this is maybe a little bit more complicated, but the numbers here, 3333, mean they carry the wild type allele at all 5 loci. If it has a 1, it carries the cultivated allele at 1 of the loci. So 1, the first 1 would mean fruit weight 2.2 is a 1. So fruit weight 2.2 does not have, on its own, does not seem to have a huge impact on fruit size. But if you get more and more fruit weight alleles in there, the fruit size increases. But the greatest increase in this population, I have to say, it's I cannot conclude that this gene does have the most impact, but the greatest increase in this population comes if the accessions carry the cultivated allele of fruit weight 11.3, and that's shown here. So in that population, 11.3 plays a big role in increasing fruit size. So we, of course, had genome sequence. We had all this phenotype data, a ton of data. So we did GWAS because we can. So we found for fruit weight the usual suspects, fruit weight 2.2 and fruit weight 3.2. And for cell size, we didn't actually find any GWAS signal, even though we expected 11.3 to be recognized, because 11.3 is definitely impacting cell size, but that didn't show up in the study. For the cell layers in the pedicarp, we found fruit weight 2.2 again, but not fruit weight 3.2, which we also know is impacting cell layer. So in all, the GWAS was helpful, but it didn't, for us, for the fruit weight aspect of this project, it didn't really help us tremendously. So we went back to the drawing board and do what I know best, perhaps, or know best what I can do myself almost, is go back to biparental mapping populations and map fruit weight QTLs that way. So we generated many populations, and we were paying attention to what we selected, and so we wanted to make sure that the known genes were not segregating, so that we would not map fruit weight 2.2 or fruit weight 3.2 again. At least that was the idea, as well as looking at development. So we had all these parameters for all these different tomato accessions, and did we predict that there was a gene functioning early in development, during ovary development or during fruit development, or were they more impacting the tissue, a certain tissue, the pericarp over the columella, etc. So we tried to make crosses that would generate the most insights at the genetic and molecular level. So we evaluated in 3 years 26 F2 populations in the field. And then we employed QTL-seq because our goals were just to map the major QTL and run with it, and not map also many minor effect QTLs. So QTL-seq, of course, uses bulk segregant analysis. So the 3 populations that we performed QTL-seq on are, this is the first one I want to share with you, and Alexis made this cross between these 2 varieties that we didn't know at that time, because the genome sequence analysis was not completed yet, but they had the largest difference in fruit weight, yet they carried, both accessions carried the wild type allele of all 5 fruit weight genes. And so the larger fruited tomato is here, that's BGV006868, and the smaller fruited one is 7931, is found in, I believe it's, well, Central America. And so, so this is a frequency histogram of the population. Clearly, you know, the small fruited parent is almost as small as the Pimpernellifolium, even though it's a Serraciforme. The large fruited parent is over 20 grams, and the fruit weight is clearly skewed towards that of the smaller parent. And this was the output of QTL-seq. And then we verified all these QTLs. We tried to verify all these QTLs, developing molecular markers and genotyping them in the entire population. And that showed that clearly the locus on chromosome 2, we call it fruit weight 2.3, had the most dramatic impact on fruit size. And that was confirmed following year with progeny testing. The next population are also quite distantly related. One parent is fully wild, is a Pimpernellifolium from northern Ecuador, and a Serraciforme also from Ecuador. And the frequency histogram here shows also a little bit skewing towards a smaller fruited Pimpernellifolium parent. And we identified a couple of QTLs. These ones are more centromeric, so they were not as interesting. Fortunately, the one on chromosome 1, although not highly significant, but it was confirmed in the F2 population to be a locus of interest. The last population is between two BGV0081 and a PI line from the USDA that are actually both from Peru. And they're very closely related. And now we see a more normal distribution of fruit weight. And QTL-seq results are here. They're not very good because of poor pooling at that time. But regardless, we thought there's something on 3, and indeed we confirmed that there's a locus on chromosome 3 that is quite significant. So don't go over the details of this table. I just wanted to indicate that we found these 3 major QTLs. We then analyzed them in all the other populations, whether they were likely segregating or not. And as you can see here for fruit weight 2.3, that was found to segregate in a lot of the populations that we had generated. So the rest of my talk is not published yet, and that will be about fruit weight 2.3. So many fruit weight related traits were correlated in the F2. And again, this was work done by Alexis, incredible amount of work. Alexis with a lot of help from the lab, because this was quite an intense harvesting season when you're also trying to evaluate cell layers and cell size, etc. But basically, the fruit weight traits are all quite correlated to one another. So fine mapping that was done by Lei Zhang, and in about 3 years led to this region, about 50 kb, that has a total of, I believe it's five gene models here. And then indicated here are the SNPs in the black lines, and in red are the indels. There were no big structural variants found here. So there's not, we don't expect, well, there's no structural variants, period, except for some small indels. So what's the phenotype in these nils? First of all, fruit weight in the field and greenhouse is clearly different. So again, this is in the nils now, so we're only looking at this locus and not the whole, you know, the whole parental genome. The tissue areas, pericarp and columella are increased, so it didn't really function on one tissue type. But also, quite to our surprise, is that both cell layer and cell size are increased. So this is a locus that seems to control both cell size and cell layer. And if you look at the growth as it increases from the ovary at anthesis to several days after anthesis, you can see that the increase in fruit size just gets a little bit more pronounced as the fruit grows. The fruit of the ovary itself looks different in size here, but it's the p-value is not that significant, and we couldn't actually find number of cells or the cell size to be correlated to that. So we do not know exactly yet whether ovary is truly different or not. In any case, the final fruit size is clearly different and is driven by cell layer and cell size. So let's look at the expression of these candidate genes. And so Lei took two genes just outside the integration as well, because of not outside the integration, outside the mapped region, because maybe there's some genome elements or cis-regulatory elements that are inside the fine mapped region but acting on the gene outside. So basically we see here 7 genes, and the expression level for the large fruited parent is in red and the small fruited parent is in green. And so here you see that the first gene is very high expressed, and the other genes are much lower expressed. Fruit weight 2.2 is extremely low expressed in this particular tissue type, but we can see for these 2 genes, like 290, 740 and 750 that are differentially expressed. So in 750 is much higher expressed in the small fruited parent, and the opposite is true for 740, that's higher expressed in the large fruited parent. So then, because these genes, this is the annotation again in the last genome build, and you see these genes are really closely together. So first experiment, we thought, let's do 5' race and 3' race to figure out, where one gene ends and the other begins. And then we got a little bit surprising result, actually very surprising. First of all, when Lei did a 5' race using a primer that's in the 740 gene, she actually found only read-through transcription all the way to the 750 gene in both parents. When she did 5' race with 550 alone, it's basically she found the same result, or the same endpoint, same 5' end. When she did 3' race on 740, as expected, the gene ends here. But for 750, she got, for the large fruited parent, she got only one product, which was actually, again, reading through transcription to the end of the 740 gene. And so the race results showed that there's actually differential read-through transcription, and so and leading to these 2 different gene models that are differing between the parents. One, it makes the one pair, or the one version of the gene is only 750. And the other version is actually 750 and 740 transcribed as just one gene. And so then Lei was asking the question of what, what's actually the expression level of this gene if you create a primer pair here and a primer pair on the other end for 740, what are the expression levels for these genes separately, and what is what's the expression level of this read-through transcription? And what she found kind of supports what she found by 5' race, is that the read-through transcription is much higher in the large fruited parent than in the small fruited parent, and we don't know what that means. But the other way around, if we're looking at this gene, it's clearly, the 750 is clearly differentially expressed, where it's much higher expressed in the small fruited parent than in the large fruited parent, whereas this gene of 740 is not differentially expressed between the 2 parents in the nils. So there's 2, maybe only 1 candidate that remains at fruit weight 2.3. Just a note here too, I should have mentioned it earlier, but the small fruit allele is partially dominant over the large fruit allele, and that makes the gene that's higher expressed here in the small fruited parent the more likely candidate. And that gene is encoding a helicase with a dead box. And it's very similar, this helicase, to eukaryotic initiation factor 4 alpha. And that's all we know about the function of the genes. We're actively working on transformations and knockdowns and knockouts, although knockouts often don't have a phenotype or are lethal. So we're now working on knockdowns and to see if we can figure out the function of these 2 genes that relates to fruit weight. So let's continue now, because the big question was is this a new allele? Because that was the whole quest, that we would find novel alleles. And so just to remind you, if you take the whole genome sequence, these two parents were quite distinct. But if you take, if you look at the haplotype, which is basically the 50 kb region that spans fruit weight 2.3, you'll see that the two parents are much more closely related. And they're very distinct from this clade here. And this is the clade of fruit weight 2.2. So certainly 2.3 is a distinct haplotype from 2.2. Yeah. So, suggesting that fruit weight 2.2 has been left behind, but question, of course, is it better or is it worse than the super closely linked 2.2? I hope that was clear from earlier, that 2.2 is right next to the two genes of interest. So we cannot, easily we cannot easily separate the two. So Lei set up a couple of crosses where she backcrossed the 6868 line to M82, that's a standard tomato, and made some backcrosses to M82. And this is two backcrosses and a self, and you can see that the fruit weight 2.3 allele is no different from that of 2.2. She continued one more backcross here, and maybe there's a trend, but I'm not sure. Anyway, if anything, there is no difference that we can tell between the 2.2 and the 2.2 allele, and they are haplotype, and they both function well in increasing fruit size. So with that, I want to wrap up with a conclusion slide. So one of the conclusions is that SLC arose about 78,000 years ago, and it was most likely domesticated, provided, the source of the modern tomato. It was domesticated and certainly available at markets in Ecuador and Peru and Colombia well before it became SLL, and it could have been first domesticated in Central America or in South America. That is still an open question. We see with GWAS and the biparental populations that we have evidence of novel fruit weight genes, particularly the one I showed to you today, 2.3. Some fruit weight alleles may have been left behind during domestication. That's, of course, an ongoing theme in our lab now. I've shown you that 2.3 increases pericarp and columella area. It acts throughout development, certainly after flowering and fruit development, and definitely on both the cell layer and the cell size. So my simplistic view of how fruit size can be regulated by one gene is certainly not holding up in this case. Fruit weight 2.3 is very likely distinct from 2.2 and is likely encoded by another gene. And so with that, I have one more slide, just my lab, with a picture of Alexis again, and Lei and Lara, whose work I've presented. And I think I already acknowledged the funding agency, NSF, which has been greatly supportive of my research. And I thank you very much for your time.